Hello, everybody. I'm Eleni Grosu, a PhD candidate at Radboud University Medical Center, and in this video, I will talk about the use of multispectral microscopy. Multispectral imaging can be widely used in studying the tumor microenvironment, which is a very complex multicellular ecosystem consisting of not only tumor cells, but also lymphocytes, such as the T cells, the B cells, and NK cells, myeloid cells, such as the myeloid-derived suppressor cells, macrophages, and dendritic cells, and the cancer-associated fibroblasts, to name only a few. Not only that, but in reality, the complexity of the microenvironment changes drastically between different tumor types. And on top of this, each and every of these cell populations has its own heterogeneity in terms of cell polarization, which eventually leads to changes in the secretory and expression phenotypes. We therefore need to study the phenotypical changes of all cells, both tumor and stromal cells, to be able to understand how cancer evolves and what therapeutical approaches are required to combat it. The tumor microenvironment cell composition can be studied by flow cytometry or by RNA sequencing. Still, both of these techniques are lacking the spatial context. Immunohistochemistry and immunofluorescence are also widely used and can provide information about cell localization in discrete niches, as well as cellular proximity and even intercellular contact information. Still, only 3 to 4 markers can be visualized at once, and that in the case of immunofluorescence. Now, in complex environments, such as a tumor microenvironment, unfortunately, this means very little. Here's where multispectral immunofluorescence comes into use. This technique combines the benefits of conventional immunohistochemistry and immunofluorescence with advances in multiplex staining and high-throughput imaging to offer much more information in one go. In the case of traditional immunofluorescence, all antibodies, both primaries and later on secondaries, are added to the tissue slide at once. This means that at the end of the staining cycle, you end up with primary labeling of the antigen and fluorophore-bound secondary labeling of the primary antibody. To each primary antibody, 2 to 3 secondary antibodies can be bound due to the spatial interference caused by the size of these antibodies. On the other hand, multispectral staining is a cyclic method, and in this method, each staining cycle focuses on a single antibody labeling sequence. Following primary antibody and HRP-bound secondary labeling, the thyramide-bound fluorophore covalently binds to the antibody complex as well as to the antigen itself. To be able to perform the multiplex immunostaining without antibody cross-reactivity, the antibody complex needs to be removed from the tissue before a new cycle can begin, and this is done using heat. Once the antibody complex is dissociated from the antigen, it leaves behind only the fluorophore bound to the antigen itself directly on the tissue. Thyramide-bound fluorophores are considerably smaller than fluorophore-tagged secondary antibodies and significantly brighter, meaning that more fluorophore is bound to a single antigen. This increases the fluorescence signal of all markers equally, and especially allows for signal amplification of low abundance antigens. If we want to compare between the two, multiplex immunofluorescence is 10 to 100-fold more sensitive than conventional immunofluorescence. And on top of that, the cyclic staining protocol allows the use of same species primary antibodies without any cross-reactivity, which not only reduces the optimization time, but also permits the multiplexing of up to 8 markers, otherwise impossible due to primary antibody species limitations. Combining the benefits of multiplex immunofluorescence with multispectral microscopy, we obtain significantly more information from one single tissue section. Multispectral microscopes work by selecting precise spectral bands and acquiring images for each of those bands, which are added to a multispectral cube that is used eventually for unmixing signals from one another based on predefined emission spectra. Spectral unmixing is necessary due to the overlap of fluorophore emission spectra. Here's how it works. You can see that the green fluorescence emission, which should be solely captured by filter F1, falls into a nearing filter F2. To correct for this, a program reassigns the spectral overlap to the proper signal, separating the two signals from one another. Below, you can see the spectral mixing as applied to an opal multiplex immunofluorescence panel. This figure shows the opal emission spectra and the different filters which are suitable for a 7-plex panel. Fluorophore emissions extend into nearby channels, resulting in non-specific signals. You can see it's very difficult to distinguish different markers from one another. During post-acquisition processing, emissions are reassigned to the proper channels in order to visualize the claimed signals. Here's an example of an immune 8-plex panel plus the nuclear stain DAPI, so a 9-color multispectral image. Spectral mixing removes any trace of autofluorescence and ensures clear signal separation to allow the in-depth study of the immune cell composition of the tissue. Now, in the context of a very complex microenvironment setting, such a panel could provide an enormous amount of intra- and intercellular information, as well as spatial information in one go with the use of a single tissue slice. We can even visualize intracellular co-localizing antigens. Here's an example of exactly that, where cell membrane-restricted epitopes have been fluorescently labeled all together. By focusing on the two cells appointed with white arrows in the overview picture, we can clearly distinguish that despite all markers localizing at the cell membrane, individualized and highly specific signals can be seen in each channel. Using such a clean input allows for extensive analysis possibilities. The following image was generated using the Vectra Polaris multispectral maps. The tumor tissue was scanned entirely, including the invasive zones and surroundings shown. The image was unmixed using the INFORM software to visualize markers representative of a lymphocyte staining panel. We can trace back cytotoxic T cells, regulatory T cells, helper T cells, memory T cells, and B cells based on their cell surface expression markers. In terms of analysis, in this case, one of the tiles was used for manual annotations of regions of interest. Here, we can find annotated the tumor in red and the stroma in green. Following this, trained machine learning algorithm applies the supracellular tissue segmentation and annotation to the entire image. You can clearly see in the bottom left that in the entire image, the tumor part is now colored in black and the stroma in gray. Cell annotation was performed using ImmuNet, a segmentation-free machine learning pipeline for immune landscape phenotyping in tumors. Lymphocytes were identified based on marker expression using this algorithm. Here's another example of an extensive analysis on an invasive melanoma tissue sample, in which the same lymphocyte panel staining was performed. In this case, the regions of interest, the tumor and its invasive margin, were manually selected on scanned tissues, and the lymphocytes contained in each region were again identified based on the marker expression using the same machine learning algorithm I mentioned on the previous slide. Lymphocytes that are recognized by the machine learning algorithm were exported as FCS files and were phenotyped based on inferred marker expression using a gating strategy that separated the different cell populations from one another. We see here a different way of data visualization which perfectly mimics flow cytometry while taking into consideration the spatial component. Deciphering very complex samples is very intricate, not only when it comes to post-processing and analysis, but also when it comes to the technique itself, especially deciding on the antibody choices and the staining sequence. For this reason, there are many off-the-shelf panels that have been optimized for different tissue samples for identifying specific cell populations or distinct markers which are correlated with therapeutic success or failure. For example, in our core, we already have immune checkpoint panels, myeloid cell panels, or dendritic cell panels, which can be widely used by any level users. Still, the technique remains flexible, and it offers plenty of wiggle room, especially for researchers aiming at identifying rare cell populations. To conclude, I'm going to go through some take-home messages. Multispectral immunofluorescence allows the visualization of up to 8 different antigens, even if they have the same cellular localization, therefore maximizing data collection from limited samples such as tumor biopsies or clinical specimens. The use of tyramide-bound fluorophores in the immunofluorescing staining cycle allows for signal amplification of low-abundance antigens, thereby enhancing the fluorescence signal on the tissue equally for all epitopes. Post-acquisition spectral unmixing ensures that individual target signals are differentiated from one another and allows for the subtraction of tissue autofluorescence. These two together reduce the signal-to-noise ratio drastically and lead to more accurate data interpretation. Multispectral imaging of multiplex immunofluorescence is a high-throughput method that scans whole slides, offering information not only on the tissue of interest, but also on surrounding tissue, unlike other methods such as CyTOF or Codex, which use tissue microarrays, therefore only offering very limited spatial information. This technique enables the in-depth exploration of the spatial tissue biology, facilitating the capture of spatially resolved interaction between cells. This is especially relevant in the context of oncoimmunology, where it's used as a tool to decipher the tumor microenvironment, and more importantly, as a tool with predictive capabilities for immunotherapy response, having great impact on preclinical and clinical research.